In the second video, we talked about the hydrolysis of ATP as the direct source of energy for most, but not all, biological work. And that would include muscle contraction for anything that you might be doing. We also talked about how, even with exercise, the levels of ATP in the muscle don't drop a whole lot. And here you can see some data that showing with strenuous exercises of different durations, ATP drops some, but it doesn't drop a great deal. Here's some other data that shows a similar type of thing, and even with a force reduction of about 80% due to fatigue, you can see that the ATP levels shown down here at the bottom haven't decreased that much, and so estimates show that it'll only decrease by about 40%. Well, this tells us that we must be constantly replacing the ATP as we use it, and we replace it using energy systems. And one of the energy systems, which is very fast because it's very short, is the Phosphagen energy system, which uses phosphocreatine. Phosphocreatine, like ATP, has a phosphate held onto it with a high energy bond. And so we can hydrolyze this bond and get the energy we need to go ahead and phosphorylate ADP to ATP. And this shows one example of biological work that is not powered by the hydrolysis of ATP. And so what I want you to do is pause at the video and identify the type of biological work and the energy source. I hope that this was pretty easy for you. I hope you said the biological work is chemical work because we're taking ADP and a phosphate and we're putting them together. And you should have said that the energy comes from the hydrolysis of this high energy bond. A second energy system is glycolysis, which is the breakdown of glucose. And this reviews for you how glucose gets into a cell and what happens to glucose once it does get in there. And you can go ahead and review facilitated diffusion on your own. And then again, I do want to remind you that as soon as glucose enters cells, it gets phosphorylated using the enzyme hexokinase to become glucose 6-phosphate. And then there are a couple of different possibilities or fates for the glucose 6-phosphate. If our energy levels happen to be high at the time, the glucose is probably going to be stored as glycogen. And the first thing that we do here is we go ahead and we move the phosphate from the 6-carbon to the 1-carbon, and then we go ahead and add that to the glycogen. And as we do, you can see that that phosphate is taken away. Well, if we need energy, we use phosphorylase, as shown down here, to break off a glucose molecule, but at the same time, we have to put the phosphate back on, making it back into glucose 1-phosphate. That can then be converted to glucose 6-phosphate, and then we continue on down through glycolysis. And so phosphorylase and hexokinase are both transferases because they're transferring an important functional group in energy metabolism. And here we see the steps in slow glycolysis specifically, and we know it's slow glycolysis because we end up with pyruvate at the end of it. This is a slightly older diagram, and some of the names are a little bit off. This should be fructose 1,6-bisphosphate, but I like that it shows you that this is happening twice. And so again, up here we see glucose enters the cell. We phosphorylate it to get glucose 6-phosphate. We convert that to fructose 6-phosphate. We phosphorylate it again to fructose 1,6-bisphosphate. And again, you should remember that that part of glycolysis is referred to as, and I pause to let you answer the question, as activation. And then we take the fructose 1,6-bisphosphate and we cut it in half. We break it into a 3-carbon piece with a phosphate on it and another 3-carbon piece with a phosphate on it. And this one is very quickly converted to the same thing. And now, again, because we're breaking the molecule in half, we refer to that as, and I hope that you said cleavage. And then finally, the 2 3-carbon pieces with the phosphate on it go through the same series of steps to finish this metabolic pathway. And so this finally is referred to as the common pathway. And so there we go. Right. And so I want you to look at this and identify it. And so pause the video. And I hope that you said, well, okay, this is just the common pathway for specifically slow glycolysis. And what are the end products? Again, go ahead and pause the video and write down the end products for slow glycolysis. And so you should have said that we get 2 NADH, and you can see those 2 NADHs up here as a result of that redox reaction. And we get 2 ATP. We in fact see 4 coming out, but don't forget that during activation we had to expend 2 ATP, so we're only getting a net gain of 2 ATP. 4 minus 2 would be 2. And then lastly, we get our 2 pyruvate, and so there's the 2 in front of that to indicate that we have 2 pyruvate. And then don't forget that in some cases we go through fast glycolysis, and most of the steps are the same, but we have to add a step and convert pyruvate into lactate. And again, here you can see that that's done with a redox reaction, and now the NADH is oxidized back to NAD, and pyruvate is reduced to lactate. And so again, identify the end products of fast glycolysis. Well, now they're going to be 2 NAD. Don't forget that we would have 2 pyruvate, and so we'd have 2 NADHs, which get oxidized to 2 NAD. We still get our 2 ATP. That doesn't change. And now we're going to have 2 lactate rather than 2 pyruvate as we go through that. The common pathway also has 2 examples of biological work not powered by hydrolysis of ATP. And so again, identify those and tell me where the energy source is for those to occur. Well, here, when we take phosphate and add it to ADP to get ATP, that's chemical work. And if we look at 1,3-biphosphoglycerate, oh, look at that. There is our high energy bond, again, denoted by the squiggly line. And so clearly what's happening is we're hydrolyzing that, and we get energy that's used to phosphorylate ADP. Well, again, we do that same thing down here. Another example of chemical work, and another example of phosphoenolpyruvate of a compound which has phosphate held on by a high energy bond, which can be hydrolyzed to give us that energy. So more examples of some places where biological work is powered by something other than the hydrolysis of ATP. All right, well, look at this figure and try to explain what it is that it's showing or illustrating here. So pause the video. And so as we look right here, we can see this is showing activation of glucose. And we go from glucose, which has a certain energy level, we phosphorylate it twice to get up to fructose 1,6-bisphosphate, And again, we know that fructose 1,6-bisphosphate has more energy than glucose has. And then we go through the common pathway to break this down to end up with pyruvate. Now, the one thing that I don't like about this diagram is it implies that pyruvate now is at 0 energy, and that's not the case. There's still usable energy in the pyruvate, and we're going to see later on that we continue to break it down to get energy to phosphorylate ADP to ATP. So it would be better if they indicated that this is not 0 energy at this point. There's more there. And then again, don't forget, we have one more step we can go, because this is just showing us going from fructose 1,6-bisphosphate down to pyruvate. You should recognize this as conversion of pyruvate to lactate using a redox reaction. LDH is lactate dehydrogenase. It's another oxidoreductase, which converts pyruvate into lactate. And again, the lactate is going to have a higher energy level than the pyruvate. All right, this is the Cori cycle. And so down here on the bottom, you can see how fast glycolysis is occurring in the muscle. What do we get from fast glycolysis in addition to the 2 lactates that are shown? Well, we also get a net gain of 2 ATP, as we had said before. What do we call the conversion of lactate to glucose? And the conversion of lactate to glucose is an example of what? Well, we call it gluconeogenesis. We're making new glucose from something else, and it's an example of chemical work. And again, I hope that you recognize that. We're taking two lactates and we're making them into something more complex, glucose, which in fact can leave the liver, and it probably will because we do this when we tend to be low in blood glucose. And it could be picked up by another muscle or the same muscle and used in fast glycolysis all over again. Well, since it's chemical work, what's needed to drive that? And again, what's needed to drive that is ATP. Well, look at the ATP that we get from fast glycolysis, a net of 2, and look at the ATP that's required to make lactate into glucose, 6. What do you think of the efficiency of this process? Well, clearly it's not very efficient at all, and so gluconeogenesis is just a short-term solution to low blood glucose levels. All right, well, we've already talked about glycolysis, and this is the first part of the complete breakdown of glucose, so we might refer to it as stage 1, and again, just to remind you, it is indeed happening in the cytoplasm. Well, then the pyruvate has to move into the mitochondria, and it gets converted into acetyl coenzyme A, and this is referred to as oxidation of pyruvate. The acetyl coenzyme A then goes into the Krebs cycle, and you can see that at least some redox reactions, among other things, are happening here in the Krebs cycle. You know that because we're getting NADH and FADH2s. And then finally, we move along and we go into electron transport and oxidative phosphorylation. And I will pretty much always talk about these together because in the majority of cases they are always linked. And so now we're going to go ahead and look at the continued breakdown of glucose. But before we do that, we're going to review what happens to energy in the food that we eat. And so again, as we said in the previous presentation, much of it gets stored. And unfortunately, in this country and many other countries too, but we tend to be particularly bad about it, the majority of the energy in the food that we eat gets stored. And again, I don't expect you to memorize the values that you see over here to the right, but I do want you to memorize where energy gets stored. And so it gets stored in the blood as glucose, it gets stored in the muscle and liver, really as glycogen, not as glucose, but that's okay. And then it can be stored as fat in the blood, as free fatty acids or triglycerides, in the muscle as intramuscular triglycerides, and then finally the largest store of energy in the body, far and away, is adipose tissue. And as we said before, more than 20 times the energy is in adipose tissue as in all the other sources combined. And then don't forget, we can also oxidize foods and use energy that's released to recharge ATP. So memorize where energy is stored, but you don't need to memorize the values. All right, before we move on to the rest of cellular respiration, I want you to talk a little bit about the mitochondria and identify the 4 parts that are important for cellular respiration. So pause the video and do that. All right, one part is the outer membrane, and this is indeed phospholipid bilayer, even though it's too small for us to see that here. We also have an inner membrane, and the inner membrane is also phospholipid bilayer, so to an extent this might be a little bit like the nucleus, only there are no connecting pores that connect the cytoplasm to the very inside of the mitochondria. And then we have the very, very inside of the mitochondria, which is the matrix. It tends to be watery and is kind of like the cytoplasm, but it doesn't have quite as much water as the cytoplasm, and so it's a little bit more gel-like. And then finally, we have the space that is between the inner membrane and the outer membrane, which is just called the intermembrane space. And here you can see another depiction of that. The outer membrane is this dark brown. Again, it's phospholipid bilayer. The inner membrane is this highly folded tan or beige part that you see here. And it's highly folded. Why do we do that in physiology? Why are things folded? And most of you probably said it's to increase the surface area. It's to increase the surface area. And then here is the matrix. They've shown it in blue because it is a little watery, but again, it's not very watery, so it's more gel-like. And then what they aren't really pointing out to us is that intermembrane space. The cristae are just the folds, and I really don't care whether or not you know that, so that's not a big deal. And then here is a third depiction of the mitochondrion for you to go ahead and look at. And here you can see a little bit more clearly that intermembrane space, which does become important. All right, so good. What is it that's being shown here? And so again, you should say right away, oh, it's a redox reaction. I know it's a redox reaction because I do see the NAD being reduced to NADH. And it's being done, that happens through the use of pyruvate dehydrogenase. And so you might guess that the substance that is participating in this is pyruvate, and it is indeed pyruvate dehydrogenase. The type of enzyme, the pyruvate dehydrogenase is at least partially an oxidoreductase, but the truth is that pyruvate dehydrogenase is actually a series of 5 complexes of enzymes, and so it's a little bit more complex than just to say that it is an oxidoreductase. You can see that it's also responsible for breaking a carbon off of the pyruvate, and that carbon is going to move into the blood, get delivered to the lungs, and you'll breathe it out. And so that's where it goes to as it happens. What's the end product? Well, I hope that maybe you remembered that the end product here is acetyl-CoA. And then finally, where is it that this is occurring? Well, oxidation of pyruvate is happening in the mitochondrion, and more specifically, it's happening in the mitochondrial matrix. And so that's where it's occurring. It's in the matrix. And this, again, is the oxidation of pyruvate to acetyl-CoA. Acetyl-CoA is really, really important. It's right in the middle of a whole lot of metabolic pathways, and so it's sometimes referred to as the common chemical intermediate. One of the things that you notice is that we can build acetyl-CoA into fatty acids. What they should have done here is they should have put an arrowhead here as well, because we break fatty acids down to get acetyl-CoA as well. The other thing that they haven't shown you is that we can break down amino acids to get acetyl-CoA. And as we've seen, we can indeed break down pyruvate to get acetyl-CoA, and that means that we can convert excess amino acids and excess pyruvate into fatty acids and triglycerides. All right, well, what is this? So this is a chemical reaction. We're taking the substrates, acetyl-CoA and oxaloacetate, and we're converting them into citrate. It's the very first chemical reaction of the metabolic pathway that we call the Krebs cycle. What is this? Well, clearly this is a redox reaction, and we're reducing NAD to NADH. NH plus and malate is being converted to OAA. This is also the last chemical reaction of the metabolic pathway that we call the Krebs cycle. And so you can see this is why we call it a cycle, because one of the end products is also one of the beginning substrates. And so that's why we call this thing a cycle. I'm wondering how many of you, as we've talked about this, have noticed that in these redox reactions, as we reduce NAD, we also get an H+. Well, if we do a whole lot of this, what happens to the hydrogen ion concentration? It goes up and up and up. And what happens to the pH? It goes down. And so again, I want to remind you that in fast glycolysis, we produce lactate. We don't produce lactic acid. And so it's not the lactic acid that is creating acidosis. It's protons that are coming from other places. And this is one of those places that they come from. And so now here you can see the whole thing. Here's the Krebs cycle. And it looks pretty hairy. But again, what is the first chemical reaction? Well, acetyl-CoA plus OAA yields citrate. And so there it is. What is the last chemical reaction? So you should look at it. And malate yields oxaloacetate. And at the same time, we reduce NAD plus to NADH. and look at all the H pluses that we see here. So you can see that there are important end products here, including 3 NADHs. Every time we go through a cycle of the Krebs cycle, we get 3 NADHs. And so we're reducing NAD to NADH. We're also increasing the hydrogen ion concentration. We go through a different type of redox reaction too, in which we reduce the coenzyme FAD to FADH2. We can do some chemical work and we can phosphorylate ADP to ATP. And then lastly, I do want to point out that we continue to break down the pyruvate, which became acetyl-CoA, and break off carbons in the form of carbon dioxide that goes into the blood, gets delivered to the lung, and you breathe it out. And so every single time you exhale, you're releasing CO2 that has come from energy metabolism, and specifically from oxidation of pyruvate and from the Krebs cycle. Well, the end products. So this is a summary of the Krebs cycle. That's very important. You have to know it. It occurs in the mitochondrial matrix in the presence of oxygen. So if we don't have enough oxygen, the Krebs cycle is going to shut down. We get 2 CO2 per cycle, or per acetyl-CoA. We get 3 NADH, we get 1 FADH2, and we get 1 ATP. And again, what's up there in the box is key, and you have to memorize it, and you have to know it. And so that's what you have to know. Again, moving on. And so again, I want to reiterate that the Krebs cycle occurred here in the mitochondrial matrix, as did the oxidation of the pyruvate. Now, what is it that we're seeing happening right here? Well, here you can see NADH or FADH come in, and they go through an oxidation reduction reaction in which the NADH is oxidized and the electrons are passed on. The yellow line shows the pathway of the electrons as they participate in a whole series of redox reactions, ultimately getting picked up by what is known as our final electron acceptor, or oxygen. You can see that we have to break molecular oxygen in half to get a single O. And don't forget, what is it that's carrying the electrons? Hydrogen. And so when we have 2 hydrogens that combine with half an O2, well, we get water. There you go. And so there's the water that we get at the very end of cellular respiration. The same thing can happen if an FADH2 comes along and it participates in a redox reaction and is oxidized to FAD. The NAD and the FAD can go back and pick up more electrons, but here again is the pathway of the electrons that came from FADH2. Now, the other thing that you see happening here is protons are being pumped from the mitochondrial matrix into that intermembrane space. And it's important that I said pumped, because that tells you 2 things. Number 1, the protons are moving uphill against their concentration gradient. All of these positive charges that you see up here in the intermembrane space represent protons. And so there are way more protons up here than there are down in the mitochondrial matrix. Number 2, it requires energy to move those protons uphill. Where is that energy coming from? Pause the video and answer that question. Well, number 1, you should have recognized that moving protons against their concentration gradient is an example of transport work. It's work. We need to put in energy. Number 2, you should remember that redox reactions involve transfer of energy. When something is oxidized, it loses energy, and some of that energy is being used to power these proton pumps and move the protons from the mitochondrial matrix up into the intermembrane space. And so the job of ETC, this is it right here. What does the electron transport chain do? It creates this proton gradient. That's it. That's the job of electron transport. Notice that in electron transport, 0 ATP is made. None. That doesn't happen until we get over here to oxidative phosphorylation. And now in oxidative phosphorylation, this proton gradient is being used to drive the synthesis of ATP. And so again, right down here, chemical work is happening. We're taking ADP and phosphate, we're putting them together. to create our ATP. That's chemical work. Where is the energy coming from to do that? It's coming from the protons flowing downhill. And as they do that, they drive this ATP synthase to make more ATP. And so we see two more examples of biological work that are driven by something other than the hydrolysis of ATP, the creation of the proton gradient, and the chemical work of resynthesizing ATP. So there you go. And here again, I should have pointed out to you at the beginning the different parts of the mitochondria, but I hope that you paid attention to them. Here's the outer membrane. Here is the inner membrane. In between is the intermembrane space where we create that proton gradient. And then here's the mitochondrial matrix, which again, as I've said before, this is where oxidation of pyruvate occurs. This is where the Krebs cycle occurs. It's also where beta oxidation occurs, which we'll talk about not in this video, but later on. And here is a different view of both electron transport over to the left. And so here's electron transport. And again, as we've said, the job of electron transport is to create the proton gradient and not to make ATP. It's the job of oxidative phosphorylation to make ATP. And that chemical work is driven by protons flowing downhill and moving from the intermembrane space back into the mitochondrial matrix. And then finally, what we see here is we kind of put the whole thing together. And so here indeed is an overview of the complete breakdown of glucose. We start with glucose entering the cell through a GLUT molecule via facilitated diffusion. And then once in here, it gets phosphorylated to glucose 6-phosphate, and then it goes through the rest of glycolysis. And obviously, you need to put in some detail here to flesh this out a little bit. You can see that the pyruvate then has to enter the mitochondrial matrix where it gets oxidized to acetyl-CoA. The acetyl-CoA goes through the Krebs cycle. All the NADH, including the NADH from glycolysis, which has to enter the mitochondrial matrix, will go through electron transport to help create the proton gradient. And then finally, in oxidative phosphorylation, we drive the resynthesis of ATP. And so there it is. It's all put together for you. And later on, we'll talk about some examples of when electron transport and oxidative phosphorylation aren't coupled. One example is brown adipose tissue, which is an interesting thing. We'll talk about it later. But that brings us to the end of this video, and we'll move on from there.